that if you go through the rest of the steps, pyruvate oxidation, citric acid cycle, and oxidative phosphorylation. If you're only doing fermentation, this is it. You get 2 ATP. That's it. So all of that energy left in pyruvate, you don't use if you're fermenting. So oxidative respiration was great because it allowed cells to then use the energy left over in pyruvate. So if there is oxygen present, pyruvate enters the mitochondria. If there is no oxygen present, pyruvate does not enter the mitochondria. So even in your own cells, if you deprive oxygen from your cells, pyruvate will accumulate in the cytoplasm. It will not go to the mitochondria. There's no point in sending it into the mitochondria. So the first thing that happens is that you have to get it through. Now pyruvate has a negative charge. It's not going to cross the two membranes of the mitochondria on its own. So you need some kind of a passageway through the lipid membrane. The outer membrane has a channel for pyruvate. The inner membrane actually has a symporter. So it's a proton pyruvate symporter. So there are differences in the concentration outside the mitochondria, in the intermembrane space, and inside the mitochondria. So that you can get this first step through passive diffusion through the channel, and the second step is through symporter with a proton symporter. Then when pyruvate enters into the mitochondria, you need to oxidize it. So you're going to get rid of one of the carbon groups. So what happens here in pyruvate oxidation is that you break down pyruvate into a 2-carbon compound and a carbon dioxide. So you lose a carbon as a carbon dioxide. The other 2 carbon compounds are attached to coenzyme A, and you form the acetyl coenzyme A. Coenzyme A is recycled. So coenzyme A picks up 2 carbons here, then delivers 2 carbons somewhere else, then goes back and picks up 2 carbons and delivers them again. So coenzyme A is recycled. The whole point is that you started with 3 carbons for pyruvate, you lose 1, and then you use the other 2 in the citric acid cycle. The net effect is you make a little bit of an ADH. So for each pyruvate molecule, you make 1 ADH. Each glucose gives you 2 pyruvates. Each pyruvate gives you 1 ADH and 1 acetyl-CoA. So we are now at this step, pyruvate oxidation, some release of energy, you capture that in the form of NADH. Still a lot of energy left in acetyl-CoA. That's where the citric acid cycle comes in. Yes, ma'am? Excuse me? It is in the mitochondrial matrix. Plants have mitochondria also. There are eukaryotes. All eukaryotes have mitochondria, but they can use that have mitosomes and hydrogenosomes. Coenzyme A is cycled. Acetyl-CoA is produced here through the oxidation of pyruvate, and then acetyl-CoA donates the 2 carbons to the citric acid cycle, and then the coenzyme A is recycled. So the CoA is recycled, not the acetyl part. The acetyl part is you pick up 2 carbons here, you deliver them somewhere else. Any other questions? Okay. So at this point, we're down to here. So some energy has been harvested out of the pyruvate, but you still have a lot of energy stored in acetyl-CoA. And the rest of the energy steps here that are diagrammed are part of the citric acid cycle. So in the citric acid cycle, also called the Krebs cycle, there are 8 steps. Each of them has its own enzyme, and what matters is what you put in and what you get out. So if you take a look here, we start with acetyl-CoA, which is bringing 2 carbons from pyruvate. We have a 4-carbon of xyloacetate, that's the acceptor molecule. So you have 4 carbons here. You take the 2 carbons from acetyl-CoA to form a 6-carbon citrate. The CoA is now recycled. Citrate is then changed into isocitrate, so that's just an isomerization. And then in the next step, you lose a carbon as a carbon dioxide. So now you go down to 5 carbons. So alpha-ketoglutarate is a 5-carbon compound. Now that process releases a little bit of energy and you harvest that in the form of an NADH, which means that's a redox reaction. Next stop, alpha-ketoglutarate to succinyl-CoA. So again, CoA is used and it's going to be recycled again. Alpha-ketoglutarate has 5 carbons, succinyl-CoA has only 4. So you're 4 carbons attached to the CoA. So you're losing a carbon here again. And then you're harvesting some of the energy in the form of an ADH. For the rest of the cycle, the 4 carbons don't go anywhere. So you're not going to lose any or gain any more carbons. So you started out with 4 carbons with oxaloacetate, you end with 4 carbons with succinyl-CoA, and the 4 carbons are recycled. Now in the next step, it's interesting because you're going from succinyl-CoA to succinate, so you're releasing the coenzyme A here, and this step makes it GTP. Doesn't make an ATP, it makes it GTP. And the GTP is then used to make an ATP. Both of those are substrate level phosphorylation. So again, no oxygen requirement here. So you're transferring the phosphate group from this to GDP to make GTP, and then transfer the phosphate from GTP to ADP to make ATP. So far, so good. Yes, sir.
This reaction is happening because the carbons are having like covalent bonds. Is that how they're losing it?
You are rearranging the carbons here, and I'm going to show you some more diagrams. The whole cycle starts because you're bringing 2 carbons from pyruvate in the form of succinyl-CoA.
What's the attraction to that? What is it on?
There is an enzyme here that is going to carry out a reaction between oxaloacetate and acetyl-CoA to make citrate. So each of these steps has its own enzyme.
And the isocitrate, how does it lose a carbon to carbon dioxide?
So isocitrate, the AT tells you it's an organic acid, so it has a carboxyl group, which is COOH, and that's the carbon you cut off, which comes off as a carbon dioxide. And then the other 5 carbons are left in alpha-ketoglutarate. Same thing happens when you go from alpha-ketoglutarate to succinyl-CoA, you lose another carboxyl group as a carbon dioxide. I can hear you.
Why can't you just go automatically from ATP to ATP? Why does it go through?
Why does this enzyme use GDP instead of ADP? Because that's what it does. Yes, sir.
But how does this release free energy?
Sorry, say that again?
How does this release free energy?
Well, some reactions require an input of free energy, some of them release energy. So when you start with something bigger, like a 6-carbon compound, and then you break it down into smaller and smaller molecules, that tends to release energy. So that's a catabolic set of reactions as opposed to anabolic reactions. Yes, sir.
So both isocitrate and alpha-ketoglutarate have carbon carboxyl groups, so that's why carbon dioxide is released along with that H that goes to that carbon.
Yeah, so anytime you see an ATE, that tells you it's an organic acid which has a COOH.
How to get more likes?
And that H breaks off the COO, and that's why you have the reaction.
Do you
So
collect
that would
the water?
ionize, so you get COO minus, and then the COO can come up. And it's simplifying these reactions immensely, but for our purposes, yes.
And you know the likes?
Alright, so the rest of the steps here are just redox reactions, so you harvest a little more energy in the form of an ADH2 and a little more energy in the form of an ADH, and then you recycle the oxaloacetate that you started with. It's very important to recycle that so that the cycle can continue the next time a pyruvate is oxidized. If you do not generate oxaloacetate, you run out. So you have to generate oxaloacetate. So let's take a look at this in a little more detail. First of all, I should give a shout to the other person besides Krebs, whose name should be associated with the cycle, except his name is kind of hard to pronounce. So people usually skip his name. And how you have to say it. So there it is. So you can just say the citric acid cycle if you don't want to try to pronounce both names. We try not to say Krebs because we want to give equal credit to both people. So just have to remember that you only get one ATP per turn, but you do get an NADH and a ADH2. And these electron carriers can then be used in electron transport in mitochondria to make more ATP. So let's take a look at this diagram, a little different. Same diagram, it just now tells you what happens in each of the steps. And it actually gives you the shape of the chemical structure of the molecules. So those of you who like to see the chemical structures, you have them in here. So we're starting on the left here with acetyl-CoA. Acetyl-CoA is bringing the two carbons left over from pyruvate. Here's your oxaloacetate, and you can see that it has two carboxyl groups here, one in either end. So you add the two carbons here, you make your citrate. Then from citrate you go to isocitrate, which is just rearranging the molecule. And then you have your redox reaction there. So you go from having 3 carboxyl groups to only 2, and you lose one as a carbon dioxide. So notice that isocitrate here has 1, 2, 3 carboxyl groups. But you lose 1, so now you have 2. So 1 comes up as a carbon dioxide. You have another redox reaction, and then you lose another carboxyl group as another carbon dioxide, so you're now down to only 1 carboxyl group. And then you don't lose any of the carboxyl groups. So the rest of them are all still organic acids, but they only have 1 carboxyl group. And here's your substrate level phosphorylation, where GDP is used to make GTP, and the GTP is. And then what happens here is you have more redox reactions to make FADH2 and NADH, and I don't really care if you remember the details of these reactions because this is not a biochemistry class. But if I were to give you this diagram, you should be able to say, where are we harvesting energy? Where are we harvesting energy? Any place where NADH or FADH2 or GTP or ATP are produced, you're harvesting energy. If you can do that for me, that'd be great. If you can tell me why we need to recycle oxaloacetate. Because without oxaloacetate, there is no cycle, because that's the first reaction. It requires oxaloacetate. So if you don't regenerate that, the whole thing stops. You can tell me what the point of acetyl-CoA is here? What does acetyl-CoA do? Brings the 2 carbons, the acetyl group, which is 2 carbons from pyruvate into the cycle, and donates them here. So if you can figure out the key steps here, that would be great. Yes, ma'am. What is relevant? I have no idea what that is. What is that? Acetyl-CoA. Yeah. You have to know why oxaloacetate has to be recycled. You have to be able to point to the redox reaction and say this is a redox reaction. Yes, ma'am. What happens to the CO2 when it's released? If you're an animal, you exhale it out. If you're a plant, it comes out of your stomata, or it could be used in photosynthesis. Okay. So, as you go through the various steps here, where NADH is harvested, where ATP is harvested, which is GTP first, then ATP, where FADH2 are harvested, you're bringing down the energy level to the minimum, which is where oxaloacetate will be. And you don't go below that. So all of the energy harvested here could be used. Some of it is in the form of ATP, which could be used directly in other reactions, but if you stored it as NADH or FADH2, then you need to do something with those high energy electrons. Which brings us to electron transport, which is almost 6 o'clock. well. So we can start electron transfer today, and then you'll have some time to think about it, and then we can talk about that and more actions of inhibitors and special experiments related to that on Wednesday.